All right, AP Bio, I'm going to start the Chapter 7 respiration notes. That way, if you're not here, whatever, you need some practice, you can watch them. But this is Chapter 7 cell respiration. And so when we talk about respiration, hopefully you can remember that it's an exergonic because it releases energy, and then the photosynthesis is endergonic. So you should be familiar with these two equations. So we're going to focus on the respiration with C6H12O6 plus 6O2, right? That should probably be a plus there where there's a question mark. I'm sorry, that's a yields. 6H2O plus 6O2. So you may want to pause the video and write down, which one's being oxidized, which one's being reduced. So when we talk about respiration, we talk about energy harvesting, and energy is released as those electrons fall from those organic molecules to oxygen. Oxygen is the final electron acceptor, which is kind of important here. And so when we talk about energy harvesting, broken down into steps. So food, will eventually glucose will be converted into NADH, which will pass the electron transport chain, which will then ultimately be picked up by oxygen, and we'll talk more about that. So our coenzyme NAD plus is our electron acceptor, and so if you remember, NAD plus picks up 2 electrons and some hydrogens become NADH, and it's storing electrons. And then the NADH carries those electrons electron transport chain, and then the electron transport chain will transfer electrons to oxygen and make water and releases that energy. And so if you paid attention during photosynthesis, this idea of an electron transport chain in these carrier molecules should not be foreign. So NAD plus is the electron shuttle. So in photosynthesis is NADP. Here in respiration is NAD plus. And there's our electron transport chain, which we've talked about. So our 3 stages of cell respiration, we've got glycolysis, which literally means sweet death, pyruvate oxidation, and citric acid cycle, which is your Krebs cycle. So hopefully remember oxidation, lose electrons, oxidize. So pyruvate is oxidized. And then we have oxidative phosphorylation, which uses the electron transport chain in that process of chemiosmosis. So oxidative phosphorylation, so it's an oxidative process that's going to create ATP by adding a phosphate. So here's our overview, which we'll probably label in class. So everything that's outside of the mitochondria, right, that's in your cytosol. So there's glycolysis, where we can take glucose to pyruvate. Then that pyruvate will be converted into acetyl-CoA, which is a intermediate. The acetyl-CoA will be under the citric acid cycle, and so on and so forth. The key point I want to pass out is you need to focus on those 2 carrier molecules, the NADH and the FADH2, because ultimately they're going to be carrying those hydrogens, dropping off electrons, but those protons are going to be used to make ATP. Let's talk about glycolysis. There's glycolysis, producing a little bit of ATP. Literally, glycolysis, sugar splitting. I think death of glucose sounds more interesting, but it's believed to be ancient. This is important. It is a conserved process that all living things do. No oxygen was available, so our cells are also able to do glycolysis without oxygen. It occurs in the cytosol, and what it does is it partially oxidizes, there's that term again, oxidize glucose to 2 pyruvates. So you're taking a 6 carbon to a 3 carbon. You get 2 ATP and 2 NADH, you make some water, but there's no oxygen required. And so we talk about glycolysis, there's 2 steps. You got stage 1, which is your energy investment stage, and you have stage 2, which is your energy payoff stage. And so this energy investment stage, where you use ATP to phosphorylate compounds of glucose, and then your energy payoff stage is where that 2,3 carbon compound is oxidized. There's that term again. And then for each glucose molecule, here's the key. You net 2 ATP produced by substrate level phosphorylation, and then 2 molecules of NAD will become NADH. So what is substrate level phosphorylation? Well, you see that term substrate means enzyme. So substrate level phosphorylation will generate a small amount of ATP. And so essentially what phosphorylation is, your enzymes will transfer a phosphate to other compounds. So that's essentially how we're making ATP. ADP plus a phosphate. That ADP comes phosphorylated. You have ATP. So there's phosphorylation. So here is just an image, excuse me, to show the energy investment phase and the energy payoff phase. So there's our summary. Glucose, we got some ATP being produced, we have NADH, and we have some water, but essentially that glucose will be converted into pyruvate. So it's about stage 2, pyruvate oxidation in the citric acid cycle. So now we're entering into the mitochondria. There's our mitochondrial structure. So we have the citric acid cycle in the matrix, and then the electron transport chain going across that inner membrane. So here's just a quick overview. There's your pyruvate from glycolysis entering. It's going to cross that membrane, and then we are entering into the actual mitochondria. So pyruvate oxidation. So pyruvate, wherever these question marks are, when I converted this from PowerPoint to Google Slides, that's supposed to be an arrow. So that pyruvate will be converted, arrow, into acetyl-CoA, and acetyl-CoA is an intermediate. And then also during that process of pyruvate oxidation, you're also producing carbon dioxide and NADH. And so now let's go to the citric acid cycle. So the citric acid cycle, which is also Krebs cycle, occurs in the mitochondrial matrix. That acetyl-CoA will be converted into citrate, and that citrate will be converted. And during that process, this is where CO2 is released. So that CO2 that you're breathing out right now, that process occurred during the Krebs cycle. So our net gain from the Krebs cycle is 2 ATP, 6 NADH, 2 FADH2, which are electron carriers. That NADH and FADH2 will be used to make ATP later on. There is a little bit of ATP produced by substrate level phosphorylation again. So here's a really complicated picture of it. You do not have to memorize this. I will show you an FRQ from 2015 where they gave you all the info. So here's a summary of the citric acid cycle. Here's our inputs, 2-pyruvate to 2-acetyl-CoA. Goes through that cycle, right? And then your outputs are 2 ATP, 8 NADH, 6 CO2, and 2 FADH2. And in my opinion, even though my opinion does not matter, the NADH and the FADH2 are the most important. All right, let's talk about stage 3, oxidative phosphorylation. So there it is. So in oxidative phosphorylation, you have two processes, the electron transport chain and chemiosmosis. So the electron transport chain occurs in the inner membrane of the mitochondria, and that's important that it occurs across a membrane because you're trying to create a concentration gradient. And essentially what happens in oxidative phosphorylation, you're producing anywhere from 26 to 28 ATP by oxidative phosphorylation via chemiosmosis. Oxidative, you're losing electrons, which are tied up into those hydrogens. Phosphorylation, you're adding ATP. So this is where most of your ATP is made. So when people say that mitochondria is the powerhouse of the cell, it's not accurate because actually the mitochondria is the place in which oxidative phosphorylation occurs, which allows your mitochondria to produce ATP. So anyways, chemiosmosis, as we talked about photosynthesis, you're creating a concentration gradient of those hydrogen ions and be pumped across. They H plus is diffused through ATP, synthase, and there you go. You got your ATP. So electron transport chain we've already talked about, but it's just a collection of molecules embedded in that inner membrane. They're tightly bound protein, non-protein components, and they alternate between a reduced and oxidized state. This does not make ATP directly, right? It just eases the fall of the electrons from the food to the oxygen. And so those electrons move the electron transport chain, though, they proton pumps, right, H plus, move across intermitochondrial membrane to try to create a concentration gradient. So there you go. NADH comes in, drops off hydrogens, they're going to be forced across. FADH comes in, hydrogens dropped off, they're going to be forced across. And the whole idea is to create that concentration gradient so that then we can move into chemiosmosis, which is an energy coupling mechanism. Coupling means that you're combining two processes. So chemiosmosis, you have that proton gradient, H plus gradient, across the membrane drives cells to work. You've got that proton motive force, again, proton H pluses to perform work. And as those H pluses pass through ATP synthase, that's that proton motive force. It's causing that ATP synthase to make ATP. And so you're using electrons from the proton gradient, flow of H pluses back across the membrane to make ATP. And so here at chemiosmosis connects the ETC to ATP synthesis. I'm trying to keep this under 10 minutes. So here's the overview. We'll do this in class. We'll talk about it. You may want to pause this and kind of go through and read and jot it back down, but that's your call. Pretty much at this point, how bad do you want the A in the class? If you wanted the A, you'd copy it down. Those of you just being good average, just move ahead. All right. So let's look at the next picture. So ATP yield per molecule glucose at each stage. We got 2 ATP from glycolysis, about 30 to 32 from citric acid cycle, and 2 from citric acid cycle, 26 to 28 ATP from oxidative phosphorylation for about 30 to 32. So anaerobic respiration generates ATP using other electron acceptors besides O2. So you can do like sulfate, nitrate, sulfur, right? But obligate anaerobes cannot survive on oxygen, so they cannot use oxygen. Facultative anaerobes, right? Muscle cells will sometimes have the ability to go in between aerobic and fermentation, and E. coli are also facultative. So there's fermentation. That's the process where glycolysis and you regenerate NAD+. And so no oxygen present, you go into fermentation. If there's oxygen present, you go into cell respiration. So without oxygen, with oxygen, again, there's your fermentation, there's your respiration. And I'll have an image for us to fill in. So types of fermentation are alcohol fermentation, lactic acid. So in alcohol fermentation, pyruvate will be converted into ethanol and CO2. So things like bacteria and yeast used in brewing and baking. Lactic acid fermentation, get your pyruvate to lactate. So fungi, bacteria, human muscle cells do that. However, the lactate, the building up, doesn't necessarily cause the muscle fatigue and pain. It's actually probably ripping the muscles. Various sources of fuel. So carbohydrates, fats, and proteins can all be used as fuel because those monomers will enter glycolysis at citric acid cycle at different points, but they can still be used to make ATP. That's why those folks that go on a keto diet, they're not consuming as much carbohydrates, so then the body will convert over to ketosis and they'll use the fat. So phosphofructokinase, PFK, this is an allosteric enzyme that'll control the rate of glycolysis, and we talked about this in class. And what PFK does is that it's the enzyme, right? It's controlling the rate of glycolysis, but it's inhibited by ATP. So if you produce too much ATP, it's going to shut off that process. And so the big picture, there you go. Again, you want to pause this, jot it down, that's your call. But this is just kind of showing the overview of cell respiration. And so there's our glycolysis and citric acid cycle. Again, you can pause these if you like, draw them, write your own caption. Um, but and there's our oxidative phosphorylation. So that is it. I tried to get that to 10 minutes. It's now at 11. So again, this is just a supplement. These are the same exact notes I'll be using in class. And so hopefully you found this helpful. So this is me signing off from beautiful room 102. Almost forgot where I was at. Tiffin Columbian High School. Have a great day, and hopefully you found these notes helpful.